are being recorded. So that's just to let you know that if you ask a question or anything later, then that will be included in the recording too. So we have a couple of upcoming talks. Next week, April 27th at the usual time here, Tuesday at 1 p.m. in Panama, we'll have a talk from Ulrich Stingel at the University of Florida titled Marine Microbial Ecology from Dwarves to Giants and from the Surface to the Bottom. And then we'll have a talk on Tuesday, May 4th at 1 p.m. the following week from Jennifer Leichleiter at the Johannes Gutenberg University in Mainz. So we hope that everyone can join us for our next talks too. And as a reminder, we are in a webinar format, so it's easiest to leave questions to the end as much as possible. But if you have something that you would like to be clarified during the talk, then you can click on the Q&A button at the bottom and type your question in, and I'll keep an eye on that, Daniela, and we can pause if we need to. But otherwise, we'll have more instructions on how to ask questions at the end of the seminar. So I think those are all the announcements. So Joe, if you would please introduce Daniela for this week.
Great, it's a great pleasure to introduce Daniela. Daniela is a research associate. She's made a big investment here. She's set up this parched experiment that she's going to talk to us about today with DOE funding, Department of Energy funding. It's designed to be a minimum of a 10-year experiment. It's a rainfall redirection experiment. Just a little more about Daniela. She recently moved from a geography department at UCLA to a better resourced science department at the University of Colorado or Colorado State. I can never keep those straight.
Colorado State University.
Colorado State. Made a move to a better resourced science department at Colorado State, and escaping the LA traffic jam. And with that, I'll let Daniela take it away.
Okay, great. Thanks, Joe. Let me try to set this up so I can see the headings. I always forget in Zoom, you can barely see your own headings. Okay, so yeah, thank you so much for inviting me. I'm really excited to get to present the research that we've been doing in Panama to you all. The title of the talk is Drying and Soil Fertility Effects on Carbon Cycling Across a Panama Rainfall Gradient. So I'll be talking about some biogeographic work across the gradient, as well as the new drying experiment that Joe mentioned, Parched. Before I start, I wanted to acknowledge the people who've done a lot of the work that I'm going to be presenting. First of all, Amanda Longi Cordeiro, who's my graduate student at CSU, who came to me from a master's in Brazil. That's Amanda under the blue umbrella there. And Lee Dietrich, who's a postdoc with me at CSU, but he's been stationed in Panama for several years. And many of you probably know him. He's down in the front in the middle picture there. Also wanted to acknowledge Karis McFarlane and Alex Hedgepeth at Lawrence Livermore National Lab, who've done some of the radiocarbon work I'll present. And Nick Booskill and Stephanie Chacon at Lawrence Berkeley National Lab, who've done some of the microbial community analyses with us in the drying experiment. And then Ben Sulman, who's at Oak Ridge National Lab. You can see the DOE influence here, right? We work with a lot of national labs. And Ben Sulman, we've been working with on the modeling component, soil carbon modeling. And then I, of course, wanted to acknowledge Joe Wright, who's our host, who's my host as a research associate, and who has been doing a lot of collaboration with us, especially on the root traits and root work that we've been doing. So let's see, I can't advance my slide, Casey. Oh, there we go, okay. So I think most of the people at this talk are probably aware that there's been ongoing drying patterns in many lowland tropical forests and montane tropical forests. We've seen drying patterns. We have predictions of increased drying patterns. This is showing one study from Brazil showing increased strength and length of the dry season. A number of other studies in the past 10 years or so have also come out from Costa Rica and Puerto Rico, drying experiments and observational studies, looking at how increased length and strength of the dry season, but also reduced rainfall overall and increased periodicity of rainstorms. All of these climatic changes in precipitation are going to be affecting tropical ecosystems. So my main interest in tropical ecosystems is in carbon cycling, soil biogeochemistry, and soil ecology. So I've been thinking about mostly soil carbon cycling since I was in my early 20s. I've been very sort of enamored with the carbon cycle in soils. This is a picture of me in Puerto Rico where I did my dissertation in the canopy, one of the canopy walks there. So as we know, tropical forests, humid and wet tropical forests in particular, are the most carbon-rich ecosystems on Earth. They hold about 30% of above ground carbon in those beautiful tall canopies. And then if we imagine those canopies turned upside down beneath our feet, there's at least 1.6 times as much carbon below ground. And the deeper we look in soil, the more carbon we find. So currently we estimate about 20% of below ground carbon stocks are in the soils of humid and wet tropical forests. So the work I've been doing in Panama, I started working there in about 2012. And I sort of started with these large sort of biogeographics questions about how soil carbon works and what drives soil carbon storage across tropical landscapes. So the first part of the hypothesized framework that I've been working on and continue to work on is that tropical rainforests and relatively resource poor sites, mainly drier or infertile soil sites, have greater root biomass and production, and that this is the main contributor to soil organic carbon stocks, and that these resource poor sites have larger carbon stocks than more resource-rich sites, so wetter and more fertile sites. And the second part is that soil carbon stocks in these resource-poor sites are less vulnerable to drying, so more resistant carbon storage because of larger root biomass with more conservative growth traits and more adaptable microbial communities in these sort of less friendly conditions, soil conditions. So the way I think about the world is really sort of a simple conceptual model with soil carbon stocks right at the middle and sort of the mass balance of carbon in and out of soils. So I'm an ecosystem ecologist, and this is sort of the first way I really started thinking about the world. The natural world was in these sort of budgets and biogeochemical cycles. So I'm going to sort of organize my talk around this the simple conceptual way of thinking about soil carbon. So first of all, soil carbon stocks and sort of internal soil processes that drive soil carbon stocks, like nutrient availability and cycling, soil physiochemical properties, microbial community and activity. Those are the ones I'll be focusing on today anyways. And then that these soil carbon stocks are just a balance of the inputs and outputs. And the main inputs I'll focus on are fine roots, as you gathered from my hypothesis, thinking about biomass, productivity, morphology, and chemistry of fine roots and how those contribute to soil carbon stocks. I'll talk a little bit, actually I won't talk too much about litter fall, but we've done some work on litter fall if you're interested. But more and more of the data out there indicating that roots are really the main direct contributors to soil carbon stocks, especially in moist tropical forests where litter fall is really decomposed so quickly. It's essential to nutrient cycling, but maybe less important for soil carbon storage. And then the main output, soil respiration, and thinking about sort of the age, what drives the age or turnover time of soil carbon, looking at the age of soil carbon that's lost. So now sort of going to my hypothesized framework, I'm going to talk about how drying affects these processes across variation in rainfall and soil fertility. So as I mentioned a little bit, we have sort of a three-pronged approach. The first is doing large-scale biogeographic studies of soil carbon cycling across the natural gradients of soil fertility that exist in Panama, on the Isthmus of Panama. So it's a beautiful sort of model landscape to address these questions about rainfall and fertility. And we've done some work across the long-term 50 long-term 1 hectare plots that have been maintained by STRI for a long time. Then we'd have this new experimental drying manipulation at a subset of 4 of those sites that were finalized in 2018. And then finally, we worked to parameterize and adjust soil carbon model processes to better represent tropical forest ecosystems. Okay, so here's the map. Many of you will be familiar with some version of this. This is the Isthmus of Panama. The first thing I want to point out is that there's nearly a doubling of rainfall from the Pacific in the south to the Caribbean in the north. And in the background, all of those colors that you're seeing are different geological substrates. So there's huge geological variation, and this is linked to huge variation in soil fertility. So it's a unique sort of place because soil fertility really doesn't vary with the rainfall, which is unusual because of this sort of overarching effect of the geology. And all of those dots are the 51 hectare plots. And I took this map from a publication we had a few years ago because I wanted to highlight how soil carbon stocks don't really vary across with rainfall. So the yellow dots are the lower carbon stock sites, white are mid carbon, and black are high carbon. And so just to keep this in the back of your mind throughout the talk, there's not a clear pattern of soil carbon across the rainfall gradient. There's sort of a hopscotching across these geological formations. And then to show you where the drying experiment is, the red circles are the 4 sites where we have the drying structures, which you can see in this picture here. It's a through-fall reduction experiment where we're redirecting about 50% of through-fall off of 10 by 10 meter plots and to sort of the outside edge of trenches that go around the plots to 0.5 meter. So the sites that we ended up using are Gigante, Buena Vista, and Sherman Crane, which are in those circles. So you can see that spans about half of the rainfall gradient. And the white blobs in this case are infertile soils and black are fertile soils. And so at Buena Vista, you can see we have a paired fertile and infertile site. And there are a bunch more dots and squares on here. And this was sort of a larger set of paired fertile and infertile sites that we initially worked on a little bit and considered. And we were going to have a number more of sites, but we had to reduce our site numbers because of theft and armed poachers and some other challenges out there. But we ended up with this nice set of sites that have a pretty representative of the larger set of 50 sites. And I'll show you some of that data to show how representative they are for the things we're interested in. Okay. So getting to sort of the first part of that conceptual model, the soil carbon stocks, right? My main interest is sort of how much carbon's out there and what's driving it. So first, I'm going to be sort of really light on methods through this talk because I think a lot of you probably know a lot of these methods, but I'm happy to answer, any detailed questions about our methods. So the soil carbon stock data is really thanks to the network of open soil pits that are out there. I know these sometimes sort of seem like a hazard to people hiking through the forest, but they're really useful for going out and resampling for something new in a place where there's already a lot of data. So that's me down in one of the soil pits. And this is my lab group that I had at UCLA where we did some analyses, and a lot of these analyses were done at the Dry Soils Lab. Okay, so first I'll give you sort of the main result from our first biogeographic study. This was published in Biogeochemistry in 2018, and this is the final structural equation model predicting soil organic carbon. And we started out with something like 30 or 40 potential predictors, all kinds of above ground measures, soil chemistry, physiology, nutrient availability, all sorts of things. And the best model really boiled down to these 4 predictors of soil organic carbon. So the single best predictor was fine root biomass. And I should say these are stocks to 1 meter depth. So fine root biomass was the best predictor, positively related to soil organic carbon. And then soil clay content and rainfall were also positive predictors, but they were quite a bit weaker than fine root biomass. And fine root biomass in turn was best predicted by total extractable base cations, and this is a negative relationship. So I'm going to show you a few of the correlations just to give you a better idea of how these things are related, the sort of one-way correlations. First, I just wanted to point out, because people often ask, like, what about all the above ground measures? Well, we did look at above ground measures, above ground biomass, DBH increment, and litter fall for the sites where we had litter fall. And this above ground biomass is from Rick Condit, who's an author on this paper. And there's absolutely no relationship between soil carbon stocks and anything that we could measure above ground. So I just want to get that out of the way. Here's that positive relationship with clay content. Sort of a nice positive relationship, and this is pretty typical of what we see across terrestrial ecosystems. The more clay that you have in a soil, the more charged surface area you have to sorb organic matter. So that makes sense. And then here are those root relationships. So a positive relationship with fine root biomass. So fine root biomass alone predicts about 30% of soil carbon stocks. So that's pretty good. And then this relationship, this negative relationship between fine root biomass and total extractable bases. So the more base cation availability there is, the less root biomass there is. And this could indicate reduced investment of plants into below ground biomass because there are enough base cations around. This could also be indicative of differences in turnover times. So some of our current work, especially Amanda Cordero's work, is focusing on looking more into those mechanisms. And I'll talk a little about that. But this relationship with base cations also dovetailed really nicely with some of Joe Wright's work on Gigante, where potassium is one of had the strongest effect on fine root biomass and turnover. So cations really emerging as a strong driver of below ground carbon dynamics. All right, so those were stocks. Those are total stocks to 1 meter depth. We want to get a little bit of a better understanding of how roots are really contributing directly or indirectly to soil carbon stocks. So we did this sort of follow-up study looking at root and carbon depth distributions. So here I've just split out the most potassium poor soils and the most potassium rich soils. And this is from all of those 50 1 hectare plots. So I've taken out sort of the ones in the middle, but they're in the publication if you want to see the full gradient. And on the y-axis, what we have is soil depth. And so what you can see here is that in those potassium poor soils, there's a lot of root biomass at the surface. And actually the surface 20 to 40 centimeters, sort of 0 to 20 or 0 to 40, explains pretty much all of the variation in total root biomass. So you can see when they get down to 1 meter depth, the potassium rich and potassium poor sites sort of converge. So we have this big chunk of root biomass in the surface of the soil. And then if we look at where the carbon is, it's almost sort of an inverse pattern. So this is extractable carbon here, but it's a similar pattern for total organic carbon. Carbon was a bit better, more responsive to total extractable bases. So here I split these out by total bases, but that's again, sort of similar to the potassium alone. And what you see here is that those really carbon-rich soils where the high carbon stocks are, most of that is actually in the subsoil, below 40 centimeters. And if you look down at the extractable base rich soils, there's this sort of more even depth distribution. There's still more carbon down here in sort of this larger volume of soil, but it's more of an even depth distribution. So to summarize this first part about carbon stocks, I sort of summarized it in black text, but I'm not going to read through that. That's sort of for the benefit of anybody watching this later who wants to read a recap. And in red text, I'll put sort of my conclusions, sort of what I draw from this. So first, root biomass appears to provide a first order control over soil carbon stocks down to meter depth across these lowland tropical forest sites. And so altering root biomass may also alter soil carbon stocks. And this is sort of leading into our interest in changing rainfall, right? If roots are responsive to changes in rainfall, then that should cascade down to soil carbon stocks. And then this interesting vertical separation between where the root biomass is in the soil surface and the soil carbon storage at depth. And here I'll just mention I have a new master's student, Emily Blackaby, who's following up on this sort of deep soil carbon controls, looking more at the sort of physiochemical properties of these soils and how that's interacting with these root inputs from the surface. Okay, so moving on to the inputs. So that was sort of the box of soil carbon. Now thinking about the inputs more from roots. So I think that first sort of big biogeographic study really drew me farther into root ecology than I had ever gone before. And in fact, I had really only thought about roots as far as biomass before I had Amanda Cordero come to my lab from Brazil. And she's a really skilled root ecologist and has brought a lot of insight to the lab. So Amanda's dissertation is really focusing on changes in root production and root morphology with rainfall and soil fertility. And so she's employed sort of two main ways to get more information about what roots are doing and how they might contribute to carbon stocks. So the first shown on the left here is using our sort of traditional root ingrowth cores, which you can see that little mesh baggie in the soil, where we measure every 3 months how much new root growth has there been across those different sites. So this work is really focusing now on the 4 sites that have the through-fall reduction structures. Although I'll show a little bit more biogeographic work about roots in a bit. So she gets these root ingrowth cores, separates all the roots out, and then she gets, of course, biomass of root growth. She also scans the roots and gets a whole suite of root traits. that can be related to function. And then her second method, which you can see on the right here, is using mini rhizotron images of roots in situ. So that top image is a schemata of these clear mini rhizotron tubes that we've put into the soil that go down to 1.2 meters horizontal depth. And then there's this sort of traditional beer can top that you have to put on top of these things to keep the rain out. There I am putting the camera down the tube after making sure it's not full of water, which happened a few times and killed the camera early on. And then down at the bottom is a little time series of these images that Amanda made. So she can observe every few weeks what's happening with the roots in these microsites and combine that data with the root ingrowth core data, which are more distributed across the plots, to get some idea of what the roots are doing. So first I want to show how her root biomass data looks, and you can sort of compare this mentally to the root biomass data that I showed you before across the 50 sites, across the whole rainfall gradient. So remember, we tried to pick these 4 sites to be representative, and I think her initial sort of live root biomass data shows that we were pretty good at doing that. So here are this color scheme holds through most of the talk. We tried to all be consistent. So in blue, we have Sherman Crane. And again, this is depth distributions of root biomass. So there you can see Sherman Crane, the wettest and most infertile site, has the most root biomass in the surface. Then we have P12, which is the infertile Buena Vista site. Then Gigante, which is also on an infertile part of Gigante, and that's our lowest rainfall. And then down there in green is P13, which is our most fertile site. So these depth distributions of roots really follow the broader patterns across the rainfall gradient and made us feel pretty good about these sites that we had selected. And I'll mention that this data come from the soils that were excavated from the mini rhizotron tubes. So this data will be paired with all of her growth data from the mini rhizotron tubes. So this is sort of the initial condition of the roots before the tubes went in. Okay, so that's sort of my ending point was biomass, and Amanda's jumping off point is biomass. So I'm going to talk a little bit now about her data looking at morphology and function of these roots. And this is work that we've been collaborating most closely with Joe Wright on. And Amanda and Joe and I have some other projects looking at root traits of species, sort of comparing species level variation. So I wanted to show a few of these beautiful images from Joe that illustrate some of these differences in root traits that we're looking at. Now, I'm not going to talk about the species level data, which Amanda is still working on, but I wanted to show these to highlight the 2 root traits I will talk about today. First, specific root length. This is length per mass, or basically how long and skinny the roots are. And so in these images, you can see that there's this sort of variation across these 3 species. On the left in Alces, you see these sort of really long, wispy, skinny roots, whereas in the middle, there are these sort of shorter, fatter roots. So that's what I mean by difference in specific root length. And generally, greater specific root length is thought to be an acquisitive trait, so associated with increased uptake of water and nutrients. Then the second root trait I'll talk about is root tissue density. This is mass per volume, so sort of how dense are the roots. And this is thought of as more of a conservative trait, so greater root tissue density is usually associated with longer root lifespan, sort of more investment of biomass into roots that are going to stick around for longer. So here is Amanda's initial characterization of our 4 sites for these 2 traits, and then I've also showed average diameter here, which is one of the other traits that she gets from her root scans. So here at the surface is where we really see separation, sort of the clearest separation across our 4 sites. For specific root length, this is smaller in the infertile sites. So you can see in green there is P13, that's our fertile Buena Vista site, and that has the highest specific root length, so the longest, skinniest roots. And then when we look at root tissue density, we see sort of the opposite pattern where the more infertile sites have higher root tissue density. And I should mention all of this data is for fine roots, less than 2 millimeters diameter. Okay, and then we have some patterns here with depth. Specific root length increases somewhat with depth, and root tissue density decreases for some sites. So sort of nice characterization across the sites here, and this separation and sort of demonstration that these root traits vary mainly with soil fertility, a little bit with rainfall as well. So as I said, Amanda's also getting that data from the root ingrowth cores over time. So that was an initial characterization. And then Amanda has gone back with a lot of help from Lee and all of our team of amazing field technicians and interns, and keep collecting and collecting and collecting root ingrowth cores every 3 months. Of course, we've been on hiatus now since last March. But I wanted to show some of this data just to show how things change temporally as well. So this again is specific root length. And now these are from ingrowth cores at 2 different time points. In brown is February through June of 2018, so including the dry season. And then in blue is the next 3 months into the wet season. And so what you see for these 2 depths, 0 to 10 and 10 to 20 centimeters, is that we have this increase in specific root length during the wet season. So remember, we had greater specific root length in the fertile sites, and now we have greater specific root length in the wet season. So it seems that at these sites, specific root length is increasing when resources increase. And that's sort of interesting because the literature often shows an opposite pattern, where specific root length increases when there are not enough nutrients and roots are sort of scavenging and searching through the soil. And then here's that pattern again, where we have the higher specific root length in the fertile sites versus the infertile sites. So Amanda's still working on relating these root traits to direct measures of nutrient uptake and some other root function. But what we do have data on is root productivity. So from those same root ingrowth cores, this is the root production during those same time periods, from sort of our first dry season that the experiment was happening to our first part of the wet season. And here what we see is something that's been seen before by Joe Wright and Joe Yavid and others who've looked at root production over seasonality. But it's nice to get, see that your own sites are doing the same thing that you'd expect from other sites. And so we see this increase in root production from the dry period to the wet period. And then we also see this higher production, at least in the top 10 centimeters at the infertile site versus the fertile site. So the infertile site appears to be investing more in root production, sort of in the early wet season here. Okay, so as I mentioned, Amanda also has complementary data from mini rhizotron images. And so these give them sort of it's faster, in some ways easier for me, not for Amanda, but easier for me to get a time series, a longer time series from the mini rhizotron images. So I wanted to show you guys that, sort of following on that root production data. So first, this is for P12. This is the infertile Buena Vista site. And this is root production from 8 mini rhizotron tubes across our sites every few weeks for sort of the next time series starting in 2019 through 2020. So here we have a lot more deep soil data. So at the top, we have the top 30 centimeters, then down to 90. And what we see, the shaded regions are the dry season. So here we see sort of similar to what we just saw from our ingrowth cores, but with much more fine scale temporal measures. So reduced root growth during the dry season, and then this big increase at the surface during the wet season, and some smaller increases at depth in the wet season, and then again a decline. So we can compare that to the other 3 sites. Amanda was kind enough to really work hard to get all this data together. And so what we see is that this is sort of the typical pattern across our sites, except for Sherman Crane in blue here. You can see there's not that huge jump in root production at the surface in the wet season like there is at all the other sites. Although there is some increase in root production sort of more in the mid soil depth. And the other interesting thing that we saw, which is really nice, and collaborate some data from the Amazon, is that in the dry season, there's an increase in root growth at depth in the wettest site and in the most fertile site. So the more resource-rich sites seem to be able to sort of stimulate some root growth at depth, where maybe they're taking up some stored water during the dry season. Okay, so thinking about how are the roots responding now to our drying experiment. This is the last image I'll show about roots. These again are those first couple of time points that we had right at the very beginning of the experiment. This time I've split out the Buena Vista sites though. So here's P12, the infertile Buena Vista site, and P13, the fertile Buena Vista site. And then those same time periods that I said before, drier and wetter. And now we have our 4 control plots in blue and our exclusion, through fall exclusion or redirection plots in brown. And what we saw here early on, this is really like, while construction is not even quite done yet, this first period. And then this is right after construction finishes, we have this lower ingrowth biomass, root biomass growth in the exclusion plots. So as I said, this is early in the experiment, and we're a little skeptical, like, is this a pre-existing condition of these plots? But the fact that we have the same pattern at both of these very different sites is intriguing. And so Amanda's now working with a whole bunch of people assisting her, interns and students at CSU, to try to get all of the subsequent time points. But so far, it seems that our drying experiment is reducing root growth in the top 10 centimeters of soil, sort of both in the end of the dry season and into the wet season, and then in the early wet season. Okay. So the conclusion sort of from this input side that I would take is, first of all, drying might reduce surface root production and inputs to soil carbon. Okay, so there may be a reduced input of that large amount of root biomass in the surface, especially in those infertile sites. But possibly drying at the surface could stimulate deep root production. We have evidence for that, at least in our resource-rich sites. Then infertile sites have greater root tissue density, and fertile sites have greater specific root length. So these are very different sort of organic chemistry and organic structure of these root types. And so one thing I'm interested in is how that affects longer-term soil carbon storage. And something that we're looking at with Amanda is how plastic are these root traits at a community scale when we impose this drying experiment. So her future data hopefully will answer some of that. And then finally, drying effects on fine root morphology could alter the tissue quality of these inputs to soil carbon stocks, with subsequent effects on carbon storage and carbon loss. Okay, so getting to that sort of third part of my conceptual model, the outputs from soils, the soil respiration. I started looking at soil respiration across a subset of the 50 sites when I lived in Panama. Joe and Helena were kind enough to go on sabbatical when I moved there. And so my family and I were able to rent their house. I'm just kidding. We were actually very sad that they were leaving, but it was really nice to be able to live in their beautiful house in Gamboa. And so one of my main activities the year that I lived there from 2015 to 2016 was to go out and survey soil respiration at as many sites as I could reasonably get to every week or two. With my team who was there with me. And this happened to be one of the strong ENSO years, as many of you might recall. So we did this sort of initial survey of soil respiration across fertile and infertile sites on the rainfall gradient in that year. And then Lee Dietrich picked these measurements up again when he moved down to Panama in 2017 and has been working on getting sort of like a finer time series of soil respiration for our 4 parched sites. So just to show you the 15 sites that I was working across during that ENSO year are these sites that we sort of used to select our parched sites from. So as I mentioned earlier, we tried to have fertile and infertile sites sort of loosely paired within different rainfall ranges. So these are the sites that I was going out and surveying with my team at the time. And this is sort of the net result from that work, which was published in Biogeochemistry in 2019. So on the left, we have soil respiration, so total surface flux across all of those dates for those different sites from 2015 to 2016. And we sort of came in towards the middle end of the wet season and then measured through the end of the dry season. So what you can see here is sort of, first of all, what you would expect if you've spent a dry season in Panama. You know, things really shut down, and soil respiration is no exception. Soil respiration sort of tanks out during the dry season at most sites. But what you can also see in this figure here is that the dots, the markers in black are drier sites. The markers in white are the wetter sites across that gradient. And so there's definitely some variation, especially during the wet season, in how much soil respiration increases relative to the dry season, right? And you see some of those dry sites really increase a lot more, whereas some of the wetter sites are kind of down almost at the levels of the dry season. So I'll talk a little bit about that sort of the magnitude of seasonal variation. Then on the right here is just showing what's happening with soil moisture at those same time points that we went out and measured the respiration. And of course, that really drops off precipitously in the dry season across those sites. So thinking a little bit about that seasonal variation, the magnitude of the seasonal variation at different sites, we decided to look at the wet to dry season ratio of soil respiration. So how much higher is the wet season relative to the dry season at a given site? And that's what's on the y-axis for these images. And this was the result of a multiple regression, which started with a forward stepwise model building exercise, where again, we started with those sort of 30 to 40 different parameters that we had data for across all these sites. And these are the final 3 predictors of the magnitude of seasonal variation in respiration. Mouthful. But on the left here, what you see is that the strongest predictor was soil resin P. So a good measure of available phosphorus, we think. So the more resin P there is, the more soil respiration can increase in the wet season. So it's not just dependent on moisture going up, there also have to be enough nutrients around for roots and microbes to crank up. So that was the strongest predictor, but in the multiple regression extractable based cations were also a significant positive predictor. And then the wet to dry season soil moisture ratio. So of course, soil moisture, the amount that soil moisture changed also varied among the sites. Now, this is some of the newer data that Lee and our team in Panama has been collecting since 2017 for soil respiration at a finer scale across our 4 sites. And here I added on my data from earlier. You can see down here, this goes all the way back to 2015. And in shade is the dry season again. And here there's sort of a gap, as you can see, from 2016 to 2017 when I was having my second baby and Lee wasn't in Panama yet. We couldn't really get everything going under that scenario, but we picked up when Lee got back down there. And an interesting pattern that's been emerging from these couple years of data is that the wet season isn't uniform in terms of soil respiration. What we really see is that soil respiration peaks early in the wet season. And it's really the seasonal transition that's super important and explains a lot of the variation among sites. How much does soil respiration really pick up early in the wet season? And so this is some of the data that we've been working with the modeler, Ben Sulman, on to sort of parameterize and update some functions in his model, which I'll talk about in a minute. But so we're really interested in this and we're continuing to monitor this. And now Lee is starting to look at how the drying experiment might be influencing these seasonal patterns. So I wanted to highlight here just briefly some of the radiocarbon work that Karis McFarlane has been leading with Alex Hedgepath, and we also teamed up with Andy Nottingham. So Karis was really interested in warming and drying, and we were really interested in working with Andy and sort of seeing how the warming experiment compares with what we see in the drying experiment. So Karis came down, and Lee and an old student at UCLA who's also at Lawrence Livermore, Alex Hedgepath, helped with these measurements. And this is her apparatus for collecting soil respiration. And then she takes it back to California and looks at the radiocarbon age of the respired soil carbon. So first I'll show the results from parched, from the drying experiment. Karis made these nice graphs for me, very kind of her, and she's working on writing this data up. So on the left is just the total soil respiration. And we decided to focus one of our sampling time points on that seasonal transition period where we see that soil respiration is really ramping up. And this was also the period we thought we would have the biggest difference between our drying plots, our dry down plots, and our control plots, right? Because as the rainy season is coming in, the control plots wet up faster than the dry down plots. And we thought that would be our sort of most likely place we would see a difference. So first we have this dry to wet season transition. Karis only worked in P12 on Buena Vista, which was most similar to Andy's sites on BCI, and then Sherman Crane to get sort of a view of a wetter forest. So this is total respiration. During that dry to wet transition in May of 2019, we see there is a significantly lower soil respiration in the dry plots. The dry down plots stayed more similar to the dry season during this transitional period, whereas the control plots started to ramp up faster. And then later in the sort of end of the wet season in November and December, they're more similar. There's a little bit lower respiration in the dry plots in Sherman Crane, but things have sort of evened out by the end of the wet season when we have the lower soil respiration. And then these are her really cool radiocarbon data. So this is the radiocarbon of that respired CO2. Here again is that transitional period. And what she saw is that there's less negative radiocarbon signature in the dry down plots. And what that means is a release of relatively older soil carbon on a decadal time scale. So I won't go into the whole bomb curve and the contribution of bomb testing in the 50s and 60s to radiocarbon in the atmosphere. I know many of you are familiar with that, but happy to explain that, or you can email Karis, better yet, and she can explain it. But essentially what we need to know for our purposes is that 14C is a nice tracer of how fast things are happening on decadal timescales for sort of surface soils and trees. And so what we see is this older radiocarbon signal, or sorry, this higher, more positive or less negative radiocarbon signal is indicating losses of older soil carbon in our dry plots. And this pattern held for both of these time points that she measured. So we thought that was so interesting. We have a couple explanations that we can think of. One is that the drier plots are not getting as consistent inputs of fresh carbon from leaching down through the forest floor and leaching down through that really root-rich surface soil. So there's just less leaching inputs because there's less water. And then another sort of related explanation is that maybe the microbes, the soils are drier, especially during the transitional period. And so they just have to decompose whatever is nearby, whatever's handy, which will be some older soil carbon. And just briefly, because I'm sure many of you are wondering, how does this compare to the swelter sites, the warming plots? We thought this was really interesting. So first, again, on the left is the total respiration. Here you can see these patterns that Andy et al. Published some months ago, showing this huge increase in respiration with soil warming, which is to be expected, right? And you can see that here in this data that Karis collected, the warming plots are in pink. So there are these big increases in respiration, especially during the wet season in swelter. But what we thought was really interesting is that here during the wet season, especially, warming plots are also releasing older soil carbon. So actually a similar pattern to what we have with drying, but we think probably for different mechanisms. At this point, there's been a lot of soil carbon lost from the swelter plots. And so microbes are probably by necessity moving into sort of an older cohort of soil carbon to decompose. But Karis is still working on this data, so hopefully it will be out published and you can read all the details soon. But we thought this was really interesting, and we've all been talking about how we can get a factorial experiment with drying and warming now set up somewhere in between our sites. So the last part of this respiration component I want to touch on is the modeling work that I've mentioned that we've been doing with Ben Sulman at Oak Ridge National Lab. We've been working with his soil carbon cycling model, the CORPS model. And this is a great model. It includes carbon and nitrogen and many of the fluxes and pools of soil carbon that we had measured at our 4 sites over time. So we were really able to get into his model and parameterize it with data from these 4 sites in Panama, and also check some of the assumptions in the model against what we were seeing in the field. And so I think, and I'm working on writing this paper up, so this should also be out pretty soon, hopefully, or at least submitted soon. But one of the really interesting things for us sort of thinking about modeling world was comparing what we had seen out in these 4 forests in Panama with sort of the underlying assumptions in the model of how soil moisture should be related to soil respiration. So in the model, this orange line is showing sort of the default process relationship between soil respiration and CO2 flux. And what the model was predicting was that the optimal soil moisture for highest soil respiration should be at really high soil moisture. So all the way up here, it's sort of a 1 fraction of 1, like 100% of pore space filled with water. And so the model thought we should have our highest soil respiration way up here at super high soil moisture. But in green, you can see sort of more what we actually saw was that we had more of a peak down here, quite a bit lower soil respiration. And so Ben worked on this parameterization and this function, this moisture function in the model to update it for these lowland tropical sites and moved the optimal soil moisture for respiration way down to better match these 4 sites. So then he ran a series of drying scenarios to sort of predict what we might see in our field experiment and what we might see sort of more broadly across lowland tropical forests with drying. So this is the output from those model scenarios. And I'll focus first on this orange box on the right, because this is the updated moisture response based on our field data that he built into the model. And he ran these 3 scenarios, which you can see on the x-axis, a 15% reduction in soil moisture, or sorry, a 15% reduction overall the year, a 30% reduction, which is sort of most similar to our experiment, and then a 1-month extension of the dry season. And so this updated moisture response curve predicted that we'll have increased losses of soil carbon or bigger soil respiration for some of our sites with reduced rainfall. So especially for Sherman Crane and P13, this updated model is predicting larger annual losses of carbon with some sort of moderate drying. And then on the left, this was the default model before, and this predicted reductions in soil respiration across the board and increased soil carbon storage with drying in tropical forests. So we were really excited to see this big change in sort of model prediction when we updated the model based on this tropical field data. Okay, so just sort of my conclusions from this output section. Moderate reductions in rainfall are predicted to increase annual soil carbon losses in our more resource-rich sites. So we're still working on the field experiment, and we'll see if that model prediction holds true. And however, drying during the wet to dry season transition may extend those low soil respiration rates that are characteristic of the dry season. And Karis's data suggests that this may lead to losses of older soil carbon because of decreased connectivity in the soil for microbes. And so altogether, this sort of indicates that soil fertility is likely to modulate drying effects on soil respiration across tropical landscapes. Okay, now finally, this is the last section, and I want to sort of briefly go through some of those internal soil processes that I was talking about, thinking about how nutrient availability and some microbial community responses are happening with our drying experiment. So here, that's Stephanie Chacon over there with one of our awesome interns, Corey. Here's Edwin, who many of you probably know, who works with Jeff Hall mostly in Agua Salud, but we were lucky enough to steal him away for a sort of interim few-month period. And so we did a lot of soil extractions looking at nutrient availability, and Stephanie and Nick Buskill did some DNA sequencing to look at the microbial community. So now we're looking at those internal processes for just the last few minutes here. Okay, first I'll start with some of Lee's data. Lee Dietrich is working on writing these up, thinking about the nutrients. So how is nutrient availability changing with drying? First, this is the pattern of how resin extractable phosphorus changes over time at these 4 sites. So again, shaded is the dry season. And the first thing that jumps out is that green line, which is our more fertile site. So our more fertile site, we luckily learned, is actually more fertile, has higher phosphorus. And what you see, the main thing I want to point out here is that we see this accumulation of phosphorus in the dry season. So we're interested to see if our drying experiment also promotes some accumulation of soil nutrients. And some data that Lee is really recently working on, so this is sort of hot off the presses and hopefully going into his paper soon, is that our drying experiment does seem to be promoting accumulation of some soil nutrients. So here we have the 4 sites on the x-axis. Control plots are in blue, and the through-fall exclusion or reduction are in red. And these are those plot level sort of N=4 comparisons. So what we see at all of the sites is this trend towards increasing nitrate. This is an annual average, but this is mostly explained by changes in the dry season. So nitrate accumulation in our exclusion plots, and also resin P accumulation in some of the sites. And the interesting thing along with that is that we're also seeing declines in microbial biomass at most of our sites as well with drying. So this is microbial biomass nitrogen, microbial biomass carbon looks very similar. So some indication here that we're having an effective drying on nutrient availability and nutrient cycling, as well as microbial biomass. And so extending from this sort of coarsest level of microbial measurement, the biomass, this is chloroform fumigation, Nick Buskill and Stephanie Chacon did this really great study, which is also, I think, in preparation for submission. Doing some amplicon sequencing of the, I don't know if you guys can see the heading on this slide at all, I can't see it on mine, but this is for the 16S region. So this is looking at microbial community composition of mainly bacteria and archaea. So these are the 4 sites in these different colors. These are some plots that Nick sent me, and then our control and exclusion plots. So they came down after 9 months of through-fall exclusion, and they saw this really nice separation in the microbial communities among sites and no effect of the drying experiment. Then we sent them some more soils after 18 months, and now we're starting to see some sort of divergence of the exclusion plots. These are the dry down plots, which are starting to sort of congregate with this new drought microbiome, as Nick is calling it. So there's this drought microbiome emerging in the bacterial and archaeal community. Whereas the control plots are sort of staying distinct for those 4 sites. They also looked at, did some ITS sequencing of the fungal community and this is on the left here. So the 4 sites are, this is a bit messier, but the 4 sites are still somewhat distinct for fungal community. And there's sort of this region, this is at 18 months also. This may be this sort of region of convergence of fungal diversity. And what's shown here in this fungal biomass, this is fungal biomass in this these box plots here with the control in blue and the exclusion in brown. And what was really interesting, and that Nick and Stephanie are working this data up, is this big decline in fungal biomass with drying, which is most apparent in Sherman crane, but is significant across all the infertile sites, so Gigante, P12, and Sherman crane. So the infertile sites really seem to be having the microbial community hit hardest. I didn't mention, I think I forgot to point out, but in the bacterial community composition, P13 is also the only site that's not really changing. So the fertile site seems to be more resistant to this drying experiment for its microbial and fungal communities and fungal biomass. So summarizing this last little bit, and there's a lot more data here on these sort of internal soil processes, but just to give you a taste, drying appears to increase soil nutrients and nutrient accumulation, especially during the dry season, and reduce microbial biomass, and eventually shifted the microbial community composition, which initially appeared to be resistant to our drying manipulation, but has since then sort of shown this emergence of a drought microbiome. And these changes are likely to alter the processing of soil carbon within the soils and could be linked to changes in soil carbon storage and respiration. And so we've just been meeting with Nick and Stephanie to talk more about sort of linking those sort of fine scale community composition research with the sort of my coarser scale interests in soil carbon. So in the interest of time, I'm not going to read through. These are just sort of summarizing these results that I've been going through, but this is a slide. You can look back at it if you want to go to the recording. And instead, I'll just show you sort of my graphical cartoon conclusion. So here was my initial conceptual model. And then what happens with drying? Well, especially in the wetter and more fertile sites, we're seeing these changes, possible changes that could lead to reductions in soil carbon storage. So the resource-rich sites from our data do appear to be more vulnerable to drying in these lowland tropical forests. So possibly soil carbon stocks might get smaller. Of course, soil carbon stocks are the last thing you can ever detect a change in. It's very difficult to detect a change in those, but we are predicting that this may happen because of the changes in nutrient availability, microbial community, and biomass that we've seen. The changes in fine root inputs, potentially we expect to see a shift towards deeper root biomass, a decline in productivity with drying, and sort of denser, more conservative root traits. And these outputs in particular, we expect to see some increases in soil respiration for some of the sites, and possibly losses of older soil carbon with drying as well. So this is sort of our first year and a half of data of the drying experiment. Plus all of that biogeographic sort of background work that I had been doing before. So I'd be really interested to hear anybody's input or comments on this as we go forward. We're just starting to sort of ramp this back up as we're more and more able to get back out to the field. I know Lee has been out sort of repairing some tree falls and checking our data loggers for soil moisture and so on. So I'll just end thanking all the many people that have helped in the field and in the lab, both at UCLA, at CSU, and at the Dry Soils Lab, and then all of the collaborators.
Great, thank you so much. So I just want to let everybody know that there are 2 different ways that you can ask a question right now. So you can either click on the raised hand button and raise your hand, and I will call on you and give you permission to turn your microphone on. Or if you prefer, you can click on the Q&A button and yeah, I see you. And type your question in and I'll read it out. So I think Helena, I saw her hand first. So go ahead.
Hi, thanks. Very nice talk, Daniela. I wanted to follow up about your point about moisture. So I was just looking at Matteo Detto and Vanessa Rubio have a paper from BCI looking at variation in soil respiration using a combination of automated and manual measurements over several years. And they don't seem to have a consistent pattern of higher soil respiration early in the wet season, although they do also get that quadratic relationship with soil moisture, very similar to what you showed. I was wondering to what degree the higher values, when you do find them earlier in the wet season, just reflect basically substrate availability. That is, kind of when you look at the forest, right, early in the wet season, there's a lot of leaf litter. And by the time you get to the late wet season, it's like the ground is bare and there's nothing there left to decompose. So is the measurement really showing that the rate is lower at high moisture, or could it be basically that you're just running out of substrate? In which case, it seems like the model should treat that differently.
Yeah, I think that's a great point. And we thought a lot about sort of the cumulative substrate limitation to microbes towards the end of the wet season, because as you say, yeah, the leaf litter is sort of gone, or, aside from what's falling every day. And so we've been talking to Ben Salman about incorporating dissolved organic carbon measures there, sort of like, and there is dissolved organic carbon in the model, but it doesn't have heavy sort of weight on the respiration. So he said this would require sort of some extensive development, but he's really interested in including the substrate limitation to compare that to really, is it moisture or is it substrate? But this would require another grant, Ben said, and a postdoc for him. Definitely, okay. This is something we're definitely thinking about, I would say. I don't know, worried, thinking about.
Yeah. I mean, it does seem like the very high soil moisture, you do get oxygen limitation at some point, obviously, if you're really flooded out. But short of that, it seems like it could, the seasonal pattern could just be substrate. So it's great you're looking into that. Thank you.
Yeah, and I didn't show the that more fine scale moisture changes also. So there's the soil moisture, but the soil moisture does also slowly increase over the course of the wet season for these sites. So there is sort of like this indication that at least partially it's soil moisture.
Okay, thank you.
Great. And next, I think either Kristen or Jeff.
Yes, one of us is me. Danielle, that was fantastic. I think I have about 15 questions, so I hope you get back to Panama when we're here, so we can have some long discussions about this. And I will, just a plug, let you know that Edwin is going to be talking tomorrow on Earth Optimism thing, just for your
Oh, great. Yes, I'd love to see that.
But I guess the one question I'll ask, so if soil organic carbon is correlated with fine roots, and fine roots are really, high density to zero, say 20 centimeters or maybe a little deeper, how mobile is the carbon below that, the very deep carbon? I mean, you measure soil respiration, you're getting integrated measure from all of everything, the flux from the whole soil profile, but how mobile, because if we say that there's so much carbon in the soil, we know the soil is super deep, but how mobile is that carbon?
So by mobile, do you mean like how decomposable?
The risk of losing it and releasing it for climate change.
Yeah, I mean, there have been other studies recently showing that, all kinds of different land disturbance has a bigger impact on deep soil carbon than we previously would have thought. And so I know this is one thing Karis is really interested in, and she's gone back down and taken some additional measures at Swelter from their deep soil pits to see what's happening with the deeper soil carbon specifically, instead of sort of the integrated flux at the surface. So I think the answer is that the deep soil carbon is not as stable as we used to think it was. It's much more, it appears to be much more vulnerable to these kinds of perturbations than we had thought, which is bad news. So of course, if carbon can sorb onto deep soil clay surfaces, it can desorb from them. And this is what some other work I didn't show, but other work I did in Panama on the litter manipulation plots with Ed Tanner also showed that sort of that sorbed carbon is not as stable of a carbon pool as we once thought and as we might hope it was. So I think the deep soil carbon, if you stop having as much leachate coming through, if you have differences in warming and moisture and all these other things, or some big perturbation above ground that's changing root exudates, I think those deep soil carbon pools are not so stable, perhaps.
And could you put an order of magnitude on that? Is it like, equivalent to the surface layers, or is it?
Like how mobile it is? No. I would say it's still much less mobile. I mean, it's mostly still, more strongly sorbed and less, the perturbations are more muted at depth. I mean, unless you get out there with a backhoe or something. So I think the work from the litter manipulation plots was showing that sort of that sorbed carbon that we think of as the most stable carbon, really 5 to 10% of it is still vulnerable. So it's not like all of it is super vulnerable, but there's sort of gradations in there, and maybe 5 to 10%. I don't know, that's, just a guess.
All right, thanks. Well, we'll look forward to seeing you down here.
Yeah, I can't wait to get back down.
Great. We had I think a similar question in the chat from Stefan Schnitzer, who said, "Thanks, Daniela, nice talk. What are the ramifications for soil carbon storage for the amount of losses of older soil carbon that you're finding?" So I think you partially answered that, or maybe totally answered it. And he also asks, what is the natural background loss of older soil carbon, or is that completely recalcitrant in other conditions?
So there's been some great modeling work looking in some tropical soils and a lot of temperate soils, and Margaret Torn's group has led some of this sort of modeling, like what proportion, you get one radiocarbon number for your flux, and then you have to sort of back out what proportion of that is really, really old carbon and what proportion is newer carbon. And so I know a postdoc in Karis's lab has been working a bit on this with soil incubations. So one way to look at that, of course, is to incubate the soil for a really long time and see how the radiocarbon signal changes over time. So as you get into that more and more stable carbon. But everything that I've seen is that some older carbon gets lost, right? Slowly, like a slow drip kind of. It's not all completely locked in there. But what's interesting, I think, in Karis's data from our sites and also from the Swelter site is that even in the control plots, right, there's this mixture of radiocarbon ages being lost. But sort of maybe more interestingly is that not just at the seasonal transition when we have the biggest difference in total respiration, but also towards the end of the wet season, there's still some older soil carbon being lost from the dry down plots and from the swelter plots. So it's not like just at the time of most perturbation, it's sort of sustained beyond the the strongest moisture perturbation, if that makes sense.
Great, thanks. So I have a question. So you showed that there's increased nitrate and you said mostly during the dry season. Mm-hmm. So does that, when the rainy season starts, does that get lost? Is it leached, or is it like changed form?
Well, that is a good question. And Lee and I were just talking about how we need a really good nitrogen study to look at denitrification versus leaching of the nitrate, right? Like, where is it going? So it accumulates in the dry season, and in the dry plots, the dry down plots, it accumulates more. And then, yeah, it goes away during the wet season as it's either taken up by plants and microbes or leached away or off-gassed. And we don't know which of those things is happening without a really focused nitrogen study. But those numbers that I showed were annual averages. So even on sort of like an annual average time scale, we see that accumulation of nitrate. But this is data that Lee literally has worked up in the last few days. So I have the same question for Lee. So let's, well, and Lee's working on the time series for these data because we have a nicer time series than that. So, so yeah, stay tuned for Lee's next paper.
Great, thanks. Yeah, it's really fun to see the new results coming out.
Yeah.
Okay, I see Aaron has a hand up, so you should be able to turn your mic on now.
Hi, yeah, great talk, Danielle. A lot of interesting data and really cool experiments across the gradient. I have a question related to root physiology. You showed that there are the changes in biomass and morphology with the roots increasing in SRL and changes in root density. But I wonder how much could changes in root respiration rates potentially be contributing to variation in your outputs there in the arrow. I mean, there could be a seasonal signal in the CO2 just coming from the roots before it's allocated into the soil carbon stocks. And I wonder if there's any motivation to kind of look at that, at that loss from the roots alone.
Yeah. So actually, yeah, great question. Thank you. And this goes to some data that we haven't really worked up in great detail yet, but we are trying to separate out the microbial and the root respiration responses. So one way is these lab incubations that Karis has been running. But another thing that we've done in the field, which they also did in the swelter plots, was put in these PVC columns down to about 25 centimeters. And they have sort of holes on them with mesh that exclude different sized things. So we have a fine root exclusion mesh on one of them. We have a hyphal exclusion, like a smaller size mesh to exclude hyphae, and then a bacterial exclusion mesh, which basically is just to let sort of water flow through. And then we're using the light core to measure the soil respiration on top of each of those chambers at the same time that we measure surface fluxes. So we haven't quite gotten to working through all that data, and it's somewhat messy. We have to take account of the disturbance effect of putting these columns in. So we have sort of a control for that. We have a total exclusion column, which is just a solid PVC tube going in. But hopefully through sort of subtracting those different fluxes from each other, we will be able to back out what's the root contribution to the surface flux versus the fungal hyphal contribution versus the bacterial contribution. So we don't know yet, but we are working on it, and we're really interested in that because the surface fluxes, it's hard to know. Like we saw that phosphorus really influences the magnitude of response to the wet season, but is that because phosphorus is really important for roots or microbes or both? So yeah, we're really interested in finding out.
Right, or potentially taking some measurements on excised root tissues, across the seasonal cycles there could be useful.
Yeah, that would be a great thing to do. And I know Karis and I have talked about that and not gotten to doing it, but that would be probably a much easier way to do it. And we may end up doing that if this data, these exclusion column data are too messy to interpret alone, then I think that would be our next step.
Cool, thanks.
Great. We'll give everyone just another couple seconds to get any last questions in. So we want to thank you for the, I have another one from probably Jeff, I'm assuming.
Yeah, sorry, I'm going to be
Yeah, no worries.
not shy here since we have you. So you didn't have any dry sites. Drying, could you speculate on how drying might happen in a forest that say has 1,700 millimeters of rain, or even a drier site? I mean, what that might do?
Yeah.
We saw the response in the wet site.
Yeah, we initially wanted to include Parque Natural Metropolitano, but we were convinced by Joe that basically everything would be stolen immediately, and we actually did have things stolen, so we didn't include that. And I also scoped out some drier sites on the Azuero Peninsula where I would have loved to work, which I think are sites you're familiar with, but it was just totally unfeasible to have a site like way out there and then have all these other sites in the central lowlands. But from the sort of biogeographic study of soil respiration that we did, which included PNM, it included Cerro Galera, and Albrook, a site there by Albrook, a little forest patch. What we see generally is that there's sort of a bigger variation with drying. I should say a bigger increase with wetting at the dry sites. So when those dry sites, sort of regardless of the soil fertility, when they get wetter in the wet season, the soil respiration goes up more than at the wetter sites. And I think in part that's because of sort of what Helena was mentioning about anaerobiosis. Those drier sites get wetter, but they don't probably go anaerobic like Sherman Crane starts to get anaerobic after getting all wet up. So the dry side though, on the dry down, like during the dry season, they were remarkably similar, really across the whole rainfall gradient, but especially across the drier end of the rainfall gradient. There was like pretty stable and similar low level of basal soil respiration. So I think that they are really good, those forests, especially on the dry end, they're good at shutting down and maintaining really basic metabolism during those super dry conditions. And that year that I had those field data were from the El Nino year. So it was drier than the usual even, it stayed dry longer and it was drier. So I think unless you get so dry that the trees are really starting to suffer, dry sort of beyond their capacity to tolerate an extended dry season, I think the microbes sort of are pretty well adapted to that really dry condition. So I think you'd have to get really dry to where you start having sort of tree mortality, embolism, other problems, trees, the deeper soil water stores are disappearing. So yeah, I think we would be getting into sort of like catastrophic disturbance at that point, just from our field data.
She's saying. Thanks.
Sure.
Great. So yeah, thanks again, Daniela, for the really interesting talk and all those many results and discussion. And thanks everybody for joining today. And we hope that everyone can join us again next week at the regular time, Tuesday at 1 p.m.
Yeah, thanks so much.
Sure. See you soon. Bye.